Now, how can enzyme activity be affected? And enzymes, like everything, it's important that we are able to control them, okay? To lower their activity, to boost their activity, to maybe shut them down or turn them on if they have been shut down. And so there are some ways to do this in general and some ways to target specific enzymes. So general ways to affect enzyme activity is to change the environment. Okay, so you can change temperature, pH, and remember these are proteins, so temperature and pH have the same effect on enzymes as they would have on any protein, right? So higher temperatures might denature, lower temperatures slow it down, slow molecular motion. Okay, and all of that will have an effect on the efficacy of the enzymes. Okay, but again, temperature, pH, those are very, very general. They affect all enzymes the same way. If you want to target a specific enzyme, I just want to turn one enzyme off or one enzyme on. There are chemicals that can specifically influence enzyme activity, if you want to get a little bit more exact. Okay, so as far as temperature and pH are concerned, are kind of more general factors. Every enzyme has an optimal pH and an optimal temperature, okay, where it functions the best. Optimal conditions favor the most active shape for the enzyme molecule, and it is all, of course, about structure and function, as it always is, okay? So optimal conditions give it the best structure, and it'll have the best function. So any changes in optimal temperature, optimal pH, optimal salinity, any of those optimal conditions, it affects the efficiency of the enzyme and can change its activity. Okay. Optimal temperature for two different enzymes. So again, optimal temperature for a human enzyme would be human body temperature, 37 degrees Celsius. That is the optimal temperature for every enzyme in your body. Okay. Optimal temperature for thermophilic, thermophily means love, heat-tolerant bacteria, it's a little bit higher, 77. Okay, so our enzymes would not do well at 77 degrees, and the bacterial enzymes move too slow at 37 degrees. Okay, so optimal pH is kind of the same. So if you have an enzyme that's in your stomach, the optimal pH is the pH of your stomach, pH 2. Okay. Trypsin is an enzyme in your small intestines. Okay, the pH in your small intestines is a little bit right around 8. So the optimal pH for trypsin would be pH 8. Okay, and if you change the pH, if you put trypsin in the stomach, it would not do so great. And of course, the optimal temperature for both of these, since they're in your body, is 37 degrees Celsius. Cofactors are other things that can usually help enhance the activity of an enzyme and sometimes are necessary for the activity of an enzyme. Cofactors are non-protein enzyme helpers. Okay, so you can have inorganic, such as metal ions like calcium and sodium, potassium. Those are all inorganic cofactors. And you can also have organic cofactors, and these guys are called coenzymes. Coenzymes are vitamins and other things like that. And basically, these guys, when they're present, they help to they enhance enzyme activity. So make the enzyme function even better. ENZYM. Okay. If we want to kind of have the opposite effect and inhibit enzyme activity, this is generally want to turn it off or slow it down. Okay. We can have different inhibitors. There are two main types. One is competitive inhibitor, and a competitive inhibitor looks like the substrate. So it will bind to the active site of an enzyme and basically physically gets in the way of the substrate binding. So it competes with the substrate to bind to the active site. Another type of inhibitor is a non-competitive inhibitor. Non-competitive inhibitors bind to a different part of the enzyme, And they basically cause the enzyme or the active site to change shape, and then that renders the active site less effective. Different types of inhibitor include toxins, poisons, pesticides, antibiotics. They will all affect or shut down the activity of enzymes. And here's a little diagram that can show you how that works. So normal binding, the substrate, as you see, is perfectly shaped to fit into the active site. It binds, everybody's happy. If you have a competitive inhibitor, right here, it physically resembles part of the substrate. So it can also bind to the active site and just physically blocks the substrate from binding. Non-competitive inhibition is where the inhibitor binds somewhere else, not at the active site. But the binding of the competitive inhibitor, as you see, changes the shape of the active site. So it is no longer perfectly fit, and now the substrate can't bind to the active site because of the change in structure. Change in structure, change in function. A little bit about the evolution of enzymes to finish off here. Because they are proteins and they're coded by the genes in the DNA, if there's a change or a mutation in the gene, again, mutations aren't always bad, this can lead to changes in the amino acid sequence, the primary structure of the enzyme, which would lead maybe to a change in the secondary structure, tertiary structure, quaternary structure. Okay, and again, this is not always bad. So if we change the amino acids, that can change their substrate specificity. And this is how new enzymes evolve that can catalyze different reactions with new substrates. Okay, so under new environmental conditions, a novel form of the enzyme might be favored. So something that just kind of spontaneously happens could be a better fit for one of the reactions. Okay, and so this is just kind of looking. So here, if we change two amino acids found near the active site, change two amino acids were found in the active site, two change amino acids found on the surface. Okay, and now we have a whole new enzyme, which can have new functions and catalyze new reactions. Okay, and that's it for the first part of our enzymes. We're going to talk a little bit more about enzymes and a little bit more about a specific kind of enzyme inhibition in the next podcast.